with the predictions that we briefly discussed. Short recap. This is a particular protein structure in which you see that for KRAS, a particular protein, there is residue 12, that was a G, and now is a C. That's the connotation typically, G12 to C. And that, in fact, is a residue somewhere here. So we see two structures overlaid, and we see one residue change. Did it change, the structure? Doesn't look like changed. You do see, in fact, that, well, it's not that easy to see, but when you look in more detail, so you see the difference between the blue and the red here. More importantly, however, you see there's something bound here, and the something bound is bound differently by the two molecules. In fact, this particular variant causes cancer. So, and causes the machine to lose it, whatever the reason for that is. So sometimes these changes can be relatively subtle and have a big influence simply because it's an influence that slightly changes the way it binds. And in this particular case, it does matter. The most extreme example I showed are variants where a single amino acid change, SAF, SAV, completely changes the shape and leads to a disease here, sickle cell. That's just. And as I said, there are at this point about maybe 200 prediction methods available. The first generation methods were those that simply looked at the overall degree to which a residue that is changed, so a variant, aligns with what you see, what you expect from biophysical background features, SIFT using the Blossom matrix, or in a particular alignment, polyphen, or by looking also at the structure. Those are sort of the simple methods. And the second generation methods, they simply require to collect more data. More data simply means you need hundreds or thousands of data points. For thousands of experimental data points, then you can begin to do machine learning. You can begin to do a little bit more optimization, and that leads to methods such as SNAP, SIFT2, and polyphen now also use machine learning. And even larger data sets is sort of the third generation. Dimitri begins to talk about it, but we're actually not quite there yet because the field has not, the number of data points has not increased enough. One solution I showed you is a simple neural network, an ANN, an old-fashioned neural network, where as input you use the amino acid sequence of, say, 5 consecutive residues, and you predict the change, whether the change in the middle one has an effect with 2 output units, effect or not effect. And there's evolutionary information put in here. Again, effect or not or neutral is what is predicted. There's a score, and I'm going to show you some graphs later where the score essentially, if this unit gets the full value, the score is going to be minus 100. If this gets the full value, the score gets 100. More than anything else, historical reason, you could map it to 0 to 1. Except for the point is that if you mapped it to a score between 0, neutral, and 1, effect, then the 0 would be a 0 signal. However, the signal that something is neutral is also important. And so this is, we could map it to minus 1. We decided to use integers. There's a bunch of different input features that are relevant. And so this method performs better than many other methods. There is a sort of reliability index that allows you to relate the strength of the prediction to the performance, to the estimated performance. The growing RI here means that the prediction is stronger. Green is neutral, red is effect precision. You see both curves go up. So the stronger your prediction, the more likely you're right, is essentially what it says. Now, what I'm going to now show you are some results for disease data sets. Let's begin here, disease data. So one set of disease data is a database called OMIM, Online Mammalian Inheritance in Man. And that essentially is a collection of cases such as the one that I showed you in sickle cell anemia. So diseases where the carrier with one SAF gets a disease. Do you believe this is common? Many, I mean, let's put it differently. Does anybody in this room believe they have one of those? You could have. No. So these kind of diseases are defined as if you had it, you would have absolutely known. And most likely we would all see it. Okay, there are some diseases that are sort of more hidden, but most of these diseases here are fairly radical. They are really such that many of them are lethal. Now, typically these diseases are of the type that very few people in the world have these diseases. That's why they're called rare diseases. But again, they are really such that if you are carrier of that, you will have that problem. Some of them are less grave than others. Some are as mild as you don't have hair. Again, well, actually, it could apply to several in the room. I'm sorry, I was a little bit premature here. Anyway, so when you look now at a data set of these disease variants, and now you ask these three different methods here, what fraction of these disease variants is predicted? So my question to you is, what is the best method? So what you show, essentially, what I show here is the fraction of all, you toss in a bunch of these disease variants, and I cannot even remember how many of those were, but several thousand. Let's say it's 2,000. So I have 2,000 disease variants. The assumption is that no method has used those for training. It's not true. So some of those have been used for training. Many have not been used for training, and certainly not for all methods. And I want to sort of ignore that problem for the time being. I'm going to open the window later again. Anyway, so what is the best method?
We don't have enough information because that's basically just a number of true positives on the chart.
What is missing? Somebody else. Somebody else. Sorry for interrupting you. Stanislav is right. And he sort of partially gave it away. Can somebody sort of finish that sentence, or is that too dumb? I don't believe it's trivial. So those of you who don't get it, just dare to get a guess. It's only the positives, right? What does it mean? Negatives are missing. Okay, fair enough. What exactly does it mean? What we're not seeing here is if a method would always predict effect, right? That method would be sort of close to 100% because it always predicts the effect. It's still not a good method, right? In that sense, we need somehow the other view. The other view is not that simple to get because for the disease variants, we do not have variants that do not cause disease, except we say everything else. But that is a very, very not sharp knife, so to speak, because a lot of all the others has been trained here and has some effects, right? So methods are supposed to say something about many of the others because they do have an effect. This one is only the question. Initially, let's go a step back. The methods that we have developed essentially predict the effect upon molecular function. Right? Now we test, can they detect the effect on an organism? Because these disease variants here are all defined as they have an effect on the organism, not only on the molecular function. But molecular is much bigger, the effect somehow. Can they detect that? Because essentially this is a different thing, right? And the answer seems to be yes, but the problem is we don't have the negatives and we cannot provide neutrals. Unless you sort of compare it to well-curated data sets, again, where you have neutrals, effect, and you sort of see how well or good you do on training neutrals, training effect, you overpredict, and ultimately, after a long analysis, you get the idea that most likely the others overpredict. We can actually not prove it, certainly not on the OHM data, because we don't have negatives, okay? But this seems to be caused by overprediction of some methods. Okay, here's a different view. And that different view is all the variants, so the number here was wrong, it's not 2,000, 5,661. So these are disease-causing variants in human. This is in animals. The number is much smaller because the curation is, so human is more studied than animals, as simple as that. Although a lot of the molecular biology is done in animals, on this level here of variants, this is something that is relevant for humans because simply they lead to diseases. These diseases are seen in hospitals, and that's why they are well studied. The same will not happen for animals. They will just be killed or not seen or something like that. Now, overall, the set here has a slightly higher effect predicted. What do you believe this indicates? So this is the variance in human that cause diseases, variance in animals that cause disease, yes?
Small amount of data points leads to greater overfitting probably.
Yes, so, well, one way that essentially you can say, I have not looked at the error estimate here. And it is true that this is in the ballpark of the error estimate. So I cannot strongly make that point. But there is a tendency why it could be higher. What could be an explanation for that tendency? Why would things cause more damage in animals than in people, or variants? That's a very trivial social answer. It's a totally trivial thing. Well, ultimately, what is considered a disease in an animal is just much worse than what's considered a disease in a human. If the pig that people eat, or the cow that gives the milk, they cannot see very well, or they don't have hair, this is not considered a disease. For people, that is a disease. This ultimately is the most likely the explanation behind it. When we looked at these 117, they were all really some bad diseases, and here at least somewhere in there that were sort of acceptable things. Okay, let's go beyond singles. The point now, and that again goes back to the question, we have a method, what can we do with a method? That's the idea behind it. And one idea is, I can predict, if I have variance here, I can predict the effect for the variant. But since I have the method, I can essentially run through the protein, and at every single position, I can predict all 19 other amino acids that are not there. What would be the effect if you replaced it by those, right? So a protein with n residues, you would have n times 19 possible variants, right? And you can simply predict all of those. And when you predict all of those, you can sort of create a landscape of mutability, of change, of sensitivity to change. Because essentially this is not all the changes, right? I'm still doing only individual change. I'm not doing pairwise. So I'm not picking two positions and I say I change this one and that one. I just do it one at a time, okay? But it creates somehow the sensitivity of the protein to single point mutants. And we can use that to, for instance, predict hotspots. We can simply say that if a position in a protein is very sensitive, so almost anything you do to it is going to be predicted to have an effect, then those are the kinds of positions that are in fact protein interaction hotspots. There's an example that somehow works. Here's another example, and I'm going to switch off the light for a moment here. It may still work. No. And on top all right, it doesn't quite work. Let me just explain what you could see if the slides would be better. You see there's some more red and some more blue. More blue means less sensitive to variation. More red, more sensitive. When you sort of look at the most red regions, well, here you see is a density of red, that's a binding site. What you cannot see is that this other binding site here is also a local shell of red. But ultimately, that gives you some idea. You could use this and simply plot how sensitive to change it is. What these three plots here mean, it is depending on how you normalize. So do you read off directly the output from the prediction method, or do you normalize it in some ways? And normalization is important here. We can use that then also to look at particular proteins. This is a membrane receptor here, melanocortin receptor that is related to diabetes. And those, in fact, are the 7 transmembrane regions that are sort of indicated here. And you can simply sort of predict the effect of variance at each position, and you can then sort of guess what would be the effect of variance for this. And not only that, you could sort of look at at 2 receptors that are very similar in sequence, that have a similar structure, and that from the sequence level similarity, I cannot remember what it was here, but it's above 60%, we would say they have similar function, they have similar structure. But then we see differences in detail, and they in fact are important for the way these work in people. And we see some examples where we have the same residue, or here, there's a different coloring of the same amino acid, so it's S and S in both. So we have 2 proteins that have the same residue at that position, but at that position they have a different sensitivity to change. Okay? So then although you see SS, LL, oh no, LL is not a good example here, TT here. Although you see a T and a T, this in red has a very different sensitivity to change than that one in blue. That means you can predict that the two receptors will differ at a position where they have the same sequence. And that's a unique tool. There's nothing else that we have developed so far that could do that. Because everything we developed so far would predict that the T has a certain function. And here we would predict that the T between the two has a slightly different function simply because through its environment it is more sensitive or less sensitive to change. And we believe that matters. We can sort of put that into an entire plot where red here means more sensitive. You see a lot of red, and in fact that turns out to experimentally also be true. So a lot of cases here when you change, it does matter. The green ones change do not matter. And then essentially what you can do is sort of plot these red region onto the structures that you know, onto these 3D structures, and then sort of do some rationale, and by that rationale you get to the point where you can actually, from this map of sensitivity to change plus the knowledge of the 3D structure, you can sort of get yourself to finding the important sites, the binding sites, the most relevant binding sites without knowing. So this is an example where this sort of in-hand analysis works. We cannot do that automatically, unfortunately. We cannot go from this map and use that map, still not from that map, and predict things directly. Although Maria talked to you before Christmas on the 12th of December about protein binding sites, and essentially she uses, the most important signal that she uses is exactly that input. So the sensitivity to change. Okay, now we can also go and look at large-scale experiments, and the next few minutes are getting relatively muddy. So DMS, deep mutational scanning, are experiments that now take every single amino acid or every single residue in a protein and change it. In some cases against all 19 not native one. And ultimately, this is too simple. One way to now look at the data, we have, in this particular case, I believe in the ballpark of 20, I can't remember exactly how large the data set is here. And we plot the AUC, or you show the AUC curves, false positive, true positives, and we have a bunch of different methods. So this is a method that works relatively well if we want to predict the actual value, but just overall effect or not effect, the AUC is relatively poor. This is the one that I talked about, SIFT. This is polyphen. This is just simple conservation. So I'm just looking at an alignment and I'm asking, is that residue conserved? If it is conserved, some threshold. If it's conserved, then I call it effect. But ultimately is a little bit similar to polyphen, but much simpler. Even simpler than polyphen. And for this data set, it seems to perform fairly well. So one way in which we can sort of take this message home, and we can say, well, there's a best method here, and there's a worst method. But then we look at the actual output, the actual data. And we see that actually even that issue is not easy. So I briefly mentioned that on Tuesday, the experiments have different values the way they measure. It's not easy to sort of normalize. So if I want to compare two experiments, this is already complicated because I have to massage the data. They all measure something else, okay? Massage the data and also the way the data falls. So we can say we normalize, so we have two measurements. One has a scale from -50 to +5. And the other one from 0 to 1. We normalize them to the same scale. But then what we observe is that the distributions sit very different. So on the right is effect, on the left is not effect, and what these two measurements measure has very different distributions. So this is because proteins differ. But this is also because measurements differ. And this is because this field is just beginning. We don't have clean data, and that is typically what you see in bioinformatics. Now, in this particular case, we just map it simple-mindedly to a scale from 0 to 1, where 1 is deleterious. And we look at some subset of these. So some of these substitutions or variants people can see have a beneficial effect, and some have a deleterious effect. We throw the beneficial away because that sort of confuses the story even more. And we only look at the ones that are deleterious. Up here you see the distribution, okay? So most are not deleterious. So deleterious is more here. So most of the distribution sits somewhere here where in the realm of not deleterious. Okay? Now let's look at the prediction method, in this particular case SIFT. You see the SIFT distribution is completely different. So this, in the SIFT distribution, almost everything, or the highest part of the distribution, sits here, in what for SIFT is predicted as deleterious. Okay, so this is not a very good prediction at all. But you sort of can see this has a Spearman correlation of 0.23. So it's not 0. Okay? Although these distributions here appear to be fairly off, you see that some points are right. Along the diagonal, right? Some predictions are right. So now we look at the different methods. That's polyphen here. In polyphen, again, you see that the distribution is off compared to this one. But now you see it sort of, you may argue this is more agreement. We get to 2 more methods here. In this one, so for all of them, the main parts of the distribution, not for all of them, for the last one here, the main part of the distribution is right. Okay? But the last one actually has the lowest Spearman. So what is the best method? And we have sort of sliced this data in many ways. And ultimately, we have not found an answer. So essentially, no matter what we measure, we get a different answer. And we have not, essentially this is sort of a very strange game in which you begin to sort of feed or fit, you begin to use the feature that reflects what you visually see by looking at all the data. And looking at all the data, essentially what Jonas, who did this work down here, what he did is he looked at every single protein separately. So this is an aggregation of these 20 proteins. If you look at them single-handedly, one by one, you somehow see much more data. Those are the real data, and we want to see that what we see, sort of the number that I put down, reflects what we see. And we haven't found a good number that does it. And all the numbers that sort of are making sense in terms of the way you measure performance, they all reflect something differently for different methods. So under some measure, so this is an extreme one, this method here has been optimized to get the correlation right. In fact, it was optimized on a data set like this. All the others were not. It is the only one that sort of captures the idea of the distribution, that most are not having an effect. But in terms of correlation that you would want to measure, a Spearman correlation, you see the smallest number. Yes, Stanislav?
We can view these methods as like weak predictor models.
Yes.
So have you tried, or anyone tried using something like boosting to combine these methods in like an ensemble and like use boosting or something like this?
Not really. So Dimitri has worked on something that sort of looks at a combination of methods, but not combination of methods as in these, but combination of different approaches. But ultimately this is the issue. But what I'm showing you has just been submitted in December by us, so we have sort of wrestled with these data sets for 2 years. So far we have sort of tried to come to a point that I make to you. I cannot show you a point, but that took 2 years to exact. Now it's time to somehow find a way to do all kinds of things with the data. And what you say, boosting is one, but I wouldn't directly boost because somehow all don't work. But I'm not sure where we are going. Dimitri is doing something like that as a first step. And here's some other. Here's another paper on the same story that is slightly more optimistic, because essentially by sort of avoiding to look into too many scales. So one issue that we discussed briefly about in last semester's molecular dynamics. So essentially you model the motion of protein structures. It can be very powerful, and Mark Hoffman has done that in particular when we know particular variants. And we see essentially why is that variant causing a disease, and then we see that variant has a certain of flexibility, and maybe if it would be less flexible, what type of drug could we develop that fits into that binding pocket or would inhibit that kind of thing. That sort of thing you can do with molecular dynamic simulation. Here's an example for the Gaucher disease. They came up with a very good idea from that model for drug design. Here's another story about Parkinson. But now we get into a story that I will briefly come back to in a moment. So you may all know that by now we know the human genome and ultimately the Neanderthal genome, and that comes from Svante Pääbo. Janet Kelso was strongly involved in that. And when the first genome was out from Neanderthal, there was a bunch of 78 SAFs that were such that the Neanderthal had the same as chimp and had something else than we. For the entire genome of Neanderthal, there were 78. Small number. And now, well, it's very clear, right? We believe we are different than Neanderthal. So the difference must be somehow in these 78. So if we understood what these 78 do, then we would understand why we are different from Neanderthal. And this is even cooler because we don't see a lot of Neanderthal walking around. So we could sort of, essentially, if you want, simulate a Neanderthal, if we understood them, simulate as in, we would understand how certain things would differ. And this project went completely wrong because there's not a single one of those where many years after, so we started working on this, we failed to work on this, they worked on it experimentally, many people have been working on these experimentally. For very few of these 78, we have any experimental data that explain to us now, yes, that's it. For some of those, typically for instance involved in speech, for some of those there are some ideas or some explanations that would make sense, but proofs we still don't have. So we don't have from this the Neanderthal in our computer model walk around differently. Not at all. And I'm going to leave it by that. I'm just going to show you one thing that went wrong, and that was done by Arthur Dong. So Arthur Dong put these 78 onto the protein interaction network and simply asked, well, for all these 78, how do they affect this network of protein interactions? And it is a quality issue, but here you have essentially all the proteins related to these 78 and all their interactions. Right? And this is where so the interactions are these dark black lines that you see between these circles. So essentially there's absolutely nothing that we know about any of these proteins. There's no interaction known. So there's no, we absolutely have no single, we wanted to see how do these 78 change the data set of protein interactions. Zero signal. And those kind of things were the problem. And because of those kinds of things, you typically need predictions. And now I sort of get over into a different mode of what we can learn from it, a particular story. And that particular story is sort of beginning with this word individualized medicine or personalized or precision medicine as it is used today. And one way of sort of defining that is by saying, well, we now know the genome. You can determine your genome. Go to Nebula or 23andMe, you get your own genome, right? What do you learn from your genome about your diseases? And that is an issue, or is a simple question that is at this moment the question of how do we change the way we do medicine in order to gain information from that genome, okay? And ultimately, what we want is that different genotypes, so different genomes, lead to different treatments. In order to do that, we have to sort of look at many aspects of the difficulty, and it's not easy. But what we do have is the completed genome since 2000, and what we essentially now know is this entire thing contains three G letters, so 3G nucleotides. That is the genome, which corresponds to about 20,000 proteins. So we all have about 20,000. I say about because we still don't have really 20,000 in the tube. So we don't have there's nobody who has these 20,000. So we still don't have all of these proteins in a way that we can do experiments with them. Almost all of them, but not quite all of them. We assume it's about 20,000, okay? Let's keep it there. And that's a big change because initially when the human genome was published in 2000, the assumption was it was 35,000 or something like that. So the number has come down and may shrink a little bit more. Proteins are the machinery of life. To repeat every single of my sort of 37, I have 37 trillion cells. I counted them. So there's a website you can put on your weight and your all kinds of things, and then ultimately you can get the number of cells. That entire, all 37 trillion cells have the same genome, okay? It's like these 4,000 books. It's the same information contained. In this particular case, actually it's not a joke, I counted those 4,000 books. This is a little bit Photoshop. There's a subset that I just had to repeat. But those in the subset I did count. Anyway, so this is like the thing that contains your human genome, and is every single one of your roughly 30 trillion cells. The other thing about number of cells, when you grow muscles, are you going to have more cells? Whatever is your background, grow muscles in your head, yeah?
I think you will only get stronger correlations between...
Connections between cells?
Yeah.
No, that's neurons. So you're too much of a head guy. Yes, the brain absolutely benefits from connections. The muscles not really. They gain from cells, from more muscle power. And more muscle power comes from more cells? No, actually from bigger cells. So what happens if you grow in muscles is your cells get bigger. The number of cells is not changing through your exercise. It's just the shape of the cells is substantially changing. And again, brain exercise, that leads to a physical change. Neurons change. Neurons no longer change, but the connections between the neurons change. Okay, let's get back to the question. So we know it's 20,000 genes. We may actually, by the way, have 100,000 constructs. Let's not get into that. Now the next question is, how do we differ? When the human genome was done initially in 2000, the assumption was we essentially have one. We briefly discussed that on Tuesday, and we need one reference genome, and everybody else will be defined as a differential to that reference genome. And that brings us to the question: how much do we differ? Does anybody have an idea? And what I'm going to talk about here is only the part of the proteins. So you and your neighbor, by how many amino acids, by how many SAFs, so to speak, do you differ? Yeah?
Well, if we totally have about 3 billion...
That's nucleotides. We have, that's an important, let me explain why that is an important difference. And now everything is gone. Oh. Somebody is boycotting the idea that I write down in a way that nobody can understand. Nobody seems to have a pen, a marker in their pocket, a not permanent marker. No. So, okay, the 3 billion is nucleotides. 97% of those 3 billion is not expressed. It leads to 20,000 proteins. Again, roughly speaking, you have protein 500 residues long. 20,000 times 500 is roughly 10 million. So you have 10 million. So it could be, the answer could be maximally 10 million, and minimally 0. So where's the answer?
About 100,000.
So that is actually a number that is very close to the real thing. I don't know where you get that from. So mapped onto the Bavarian coat of arms here, blue is where you and your neighbor have the same, red is where you differ. So in fact, it's 20,000 differences to your neighbor, roughly. And 20,000 differences, Stanislav, when initially the human genome was done, was way more than people thought. Initially, the impression was that typically when we are related people, we differ by only some, not 20,000, but maybe 20. That was more sort of the idea. So this is in fact a rather big number. And again, why is it a rather big number? Remember, every one of my 37 trillion cells has the same genome, no difference. So this is a lot of difference. Okay, now the next question obviously is, do they have an effect? So we are talking about people here who were healthy as defined by the fact that they walked into a place and could do their genome. So we are all healthy, we are all here. So that is one. So none of us, let's put it very simply, none of us suffers from a disease that killed them before they were born. Okay? It's a very simple definition. It's an extreme definition of healthiness. And actually, health insurance would be much cheaper if that would define health. But anyway, let's not get into that. They are clearly not really killing or not before being born kind of diseases. And so how would you answer the question, on average, what is the effect of a variant? Or somehow, do many of them have an effect? How would you answer that question? So now I know it's 20,000. Now I want to know what's the effect of those. I assume they don't have a big effect because we can all walk around. I assume they have some effect because somehow we look different. Yes?
No, like you just said that we all look different. So it is an effect, it's just not an effect that influences our...
No, but that would, so for instance, hair color.
Yeah.
Those are effects that we can see on the genetic side. We have differences in the genes. But do they up to, so if I have the 20,000, how many of those would actually be the difference in the way we look? And how many of them would be neutral, yes?
What's the number for example twins? Is it also?
0.
0.
2 twins is 0. Another important number here for you is if I take both genomes of your parents and take yours, and I ask, so any difference that you have, you may have a difference to your mother or to your father. But I say, how many are new? So how many are such that neither your mother nor your father has it? And that number is less than 3. Not 3,000, really 3. Okay? Twins are 0. Again, depends on what time. Sorry, I was a little bit too fast. Depends on the type of twin. But if they come from the same cell, 0. Because that is the same genome. And again, with respect to both your parents, you have only 3. So that's a tiny difference. Yes? And you do look different from your parents. This is sort of a point where this idea, every single one, or we need a lot of changes in order to make us look different. May not be. Right? Okay, so what would you do? Well, one way would be to simply ask, for how many of those do we have experimental results? I talked about a data set that we use for machine learning with 40,000. I talked about these 5,000 or 5,661 OMIM variants. So we have a lot of variants that we have experimentally characterized. But for this data set that we have, this is a tiny, 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 tiny subset. It's not even 0.1 per million, per mil. So that's the fraction for which we know something experimentally about that protein, not about the variant, about that protein. So the set, because the space that we have is so huge, the experimental data we have is relatively little. Okay? So if we wanted to get an answer by experiment, we would have to wait. I write here by 2050. We don't know. But so I gave this talk first 5 years ago, and then I wrote essentially the same slide, and the number has hardly changed because the goal is so far away. Maybe we get there by 2050, but the more we go over so extrapolating from the last 5 years, this number should be higher. Anyway, what we can do today is by computers, and that essentially is the method from Jana Bromberg and Max Lecht, where we can predict effect neutral snap. And what I'm going to show you is a bunch of slides that use this snap scale. So strongly predicted effect, strongly predicted neutral, and I'm going to have cumulative curves. Cumulative curves, essentially what I mean is, I ask what fraction of my data set is right of the threshold. Okay? And if I now threw into my method, if I would have a very good method and I threw in a data set of experimentally effect, then it should be that sort of most of those are predicted very strong effect, right? If I threw in neutral, most of those should be predicted very strongly neutral. That would be a good method. Now, how good is my method in real life? So these are 2 data points. In real life, this is experimental effect, this is experimental neutral. So way, way worse than a very good method. So this is cross-validated, the cross-validation result. The difference between the red and being up there is the mistake, or the green and being down there is the mistake. And the difference between these two is the power of the method, right? Power, because if green and red would be the same, the method would essentially have a random prediction for any variant. The fact that green and red differ means it can predict. And now you may sort of look at this and say, actually, the power is much less than the mistake. That's the best method, at least for many years, that we claimed available. Now, the first thing we looked at, and I briefly already discussed this, in orange you have the OMIM dataset. The Almond Dataset in orange here is a dataset where not a single one of them had been used for development. Is there anything that comes to your attention or anything you stumble over? If I show you this. So red is cross-validation effect. Green is cross-validation neutral. OMIM is a new dataset that I've never seen before. And it's higher.
So our method better distinguishes more stark or more drastic difference. For example, like as far as I understand, orange is a disease, so where like your phenotype is significantly different from the norm. So our method better recognizes situations where you might die, which is kind of good.
So you're totally right, that is sort of an interpretation, but the sort of technical point that I want to make here is that usually when you do machine learning and you come with a new data set, you never do better than your cross-validation. Whatever we do, whatever developers do, typically we overestimate performance. That means that whatever new data set you come with of effect would be below the red. That's the typical observation. And there are very, very few exceptions. In fact, I would challenge that the only exceptions you find are those where the people took the wrong data set to try. It was not far enough away or something like that. But whenever I really looked at people who did a good job in finding a new data set, in our case and others, it was below. Here we have one that is above. And why, and Stanislav clearly answered that already, at least answered that in a sense that we suspect. We suspect the answer is, this is because this is a much more consistent data, because the effects are much stronger. That's why they a single one can cause disease. Now, since we know this, one point that we have to make here is for us, whether a protein sort of adopts a new function or completely loses function. Both of those are effects. We cannot distinguish them. So something that makes the protein much better or something that makes the protein lose it completely for us looks the same because it's a total change. Okay, we cannot distinguish that. But at least since we can sort of predict disease variants, so we can predict from the molecular level to the level of the organism, we could hope that somehow we can predict the effect for all the 20,000 that you and your neighbor differ. Where do you believe it will be on these curves? So will it be here? Will it be below green? Where do you believe it will be? Anybody ventures a guess?
Slightly higher than green.
That's exactly what we expected. And this is exactly not what we observed. And this is why this story took many years, because student after student was kicked out because it was not believed with those data. Ultimately, this is the fate of when you get a new discovery, the fate is that nobody believes it. That's the definition of a new discovery. There's a whole field that for years has not seen it. You're the first to see it. You're going to have a tough time. As simple as that. Now, what this curve here really is it really not close to green. It's essentially just in the middle between green and red. It's essentially almost like random. It's less than random, but it implies that there's a strong effect. So to just look at one point here, a score of 50 means strongly predicted effect, and there's 25% of the entire 20,000 that we predict has a strong effect. Now we're back to the question, are these the ones that make us look different or not? Ultimately, I will not answer that question because I don't know. But what you could now ask is, if you compared us to an earlier human, where would these effects fall? So again, I don't know anything about those 78, I already told you. And now I'm going to show you the Denisovan, so it's a little bit more than 78, and so the variants are many more than 78. But where will they fall? So we don't know how the Denisovan looked, or how Neanderthal looked, for that matter, but we assume they were a different group. So evolutionarily, they are a different group. They're not Homo sapiens. Very simple. Oops, here. So the line is choppy because at the first, when we did this, now there are more. But when we did this, there was exactly one genome done from a Denisovan. There was a piece of a finger that was found, and that way the Denisovans were defined as Denisovan, only genetically. So we know at that point, science knew about these Denisovans only through that genome sequencing. It's the first species ever that was described only by genome sequencing, and that was simply one individual, while this here was about initially 1,000. And I believe I still show you the data for 1,000 people. So this is why these curves are smoother than this one. But what you see is that the variance between us and Denisovan are less on average have an effect than the ones between us. So if we believed that many of the ones that differ are the ones that make us look different, they look more like we, in some sense, right? In this logic, yes?
But can we make this conclusion based only on one genome of one Denisovan?
No. No, but we had only one. Okay, so what would you do then? We have only one Denisovan. At that point, we had only one Neanderthal, so we could not look at a hominid. Because there was those at the point we first did that. Today we have a little bit more. We could look. Well, what would you look at next? Something living.
Like apes?
Gorilla. And I'm not entirely sure. I believe I show gorilla here. Yeah. Right, exactly. And very often in science, once you have seen, so you see something that I totally didn't expect, once you have seen it, you have tons of people who jump up and say, I knew this. For us, utterly surprising. To everybody I talked to and asked, every expert I asked to predict where the curve would go, they all predicted something else. After I showed it, they expect, yeah, sure. This is what I always assumed, right? I just asked you. Anyway, that's a longer story why we assume that this may explain it, but the problem is, here's mouse, fly, I'm not showing, is even further down. The story gets a little bit more complicated because we are looking at different proteins, partially, yes?
But okay, so the fraction makes this picture, but the number of genes that are different between not a fraction of them, but the absolute number is significantly
different.
Greater.
Totally right. So I would have to redraw this for the subset where they're all the same, and then all you will see, the tendencies will be the same, but the whole thing would be much choppier. Because you will just reduce number of data points. But you would still sort of see the same tendency of a signal. I'm not going to go into what we assume what we see is what we observe, because since we couldn't predict it, I believe explaining it after we see it is a mistake. That's why I avoid it. But many people understand. Will tell you why they expected this all the time. Yes.
But I'm sorry. It's just post factum, as you said, this makes a lot of sense because this basically says that there are some core proteins that does not change at all, in any organism, and these are exactly the proteins that we see on this graph.
Well, so the way so on this, yes. So when we have a bunch of proteins that have to be maintained between a bunch of organisms. I already told you there are about 6,000 proteins that are common to all our cells, and these are the same kind of proteins that are sort of common to mouse, and these are sort of essential things, and they have a limited space. This is one way of going, and that I believe is a right way of going. Another way of going is to understand what I'm showing. What I'm showing is sort of misleading, in a sense. Here, I show a variation within a living population. Here and here, or in between these here, what I show is the difference between a living population and a split that happened a long time ago in the past. So something that is frozen in history. And ultimately, this way of zooming in may sort of distract the way I look at it, and at some point when two organisms split, they actually have the same genome. So there things do not differ very much. They are very, very similar. And this may be exactly sort of the neutral hypothesis, and then the living population changes things. Whatever it is, what this shows here, what this exercise shows, is that a lot of what makes a difference here is not going to be the reason for why we look different. Because we could create, and again I could look at the subsets where it's the same. And we still see that one thing is there's a lot of variance between us that make a difference. So in the most extreme one here, I see that 12% have an effect that is like the disease variance. Okay, then we assume, okay, this may be true, this may be so, but actually the ones that matter, they will not happen very often. So what I mean is, if I now had two data sets, one rare variance, so those are the variants that one is only observed in one particular person, or in just a few people, we have not sequenced, so I'm not entirely sure. One data set that we used is based on 70,000 people. So the rare variants are those that are observed in fewer than 1% of these 70,000. And then there is frequent, those are observed in more than 5% of the population. And the bulk of data in terms of numbers of SAFs sits here, that's 10 million. And this is sort of 50,000, but most variants are actually here. So it's simply the way you count, right? The rare variants you observed in so few people that they don't add up to the most. But again, if I distinguish them and I say I give a number to each SAF, then that is the largest number. Anyway, what I assume is that the ones that are rare will not survive. So the ones that make the change are the ones from people that will not survive. They are rare. While what is common between many people is the one that is being selected for in evolution, and that will not have an effect. That's the idea, and that's the assumption. And of course, again, I wouldn't show you that if I would be right. This is again totally wrong. It's just the other way around. So for confusion here, I put random on top of it. You see common in green, so it's slightly below random, and I should have taken the orange out. The red one is rare. The orange is somewhere in between these two. So this is less than 1%, more than 5%, and the orange is just in between these two numbers. But the point is that I expect the red to be above the green. Clearly above the green, and it's clearly not. Okay? If anything, less than expected or more selected toward neutral is the rare variance, and we have in our common variance we have a lot of change selected. Well, maybe what that means is that in fact the variants that we have that make us different are adding up of tiny minute changes, not the big ones. So a little bit plus a little bit, and that makes us look different. That is what Jana Bromberg argues in this paper. That the weak ones may define individuality. But it may also be true, or on top it may be true. So I talked about the sickle cell anemia. Now sickle cell anemia is a disease that you have a lot in malaria infested areas. Tanzania is an example for that. There's a study section for sickle cell anemia there. Because if you have that, you have a bad disease, sickle cell anemia, but actually you're also malaria resistant. So what is good in some situation can be bad in another, or put differently, right? A bad mutation can, under some circumstances, become an advantage for a population, because it sort of may make some survive by risking something that under other circumstances is not good. So maybe some of these variants that we have here, that's the sickle cell anemia story again, maybe some of the ones that have an effect, some have a bad effect and some have a good effect. So maybe we sort of could get rid of the bad effect ones and only keep the good effect ones. And now we get back to the personalized medicine. Personalized medicine, so when we look at our diversity, somehow a treatment that is genome specific, a treatment that maybe everybody gets their own drug. Right? Could that happen? So I read my genome. I'm writing now a book in the year 2050, and there's a machine that, my watch reads my genome. My genome tells me I have a certain disease, so I have to take the pill that is being made for me. Is that going to happen? Yes?
Well, you still need to know not only your genome, but how it's expressed and epigenomics and everything.
You know too much. That's true. The story is more complicated. True. Absolutely true. But let's get the book. You're absolutely right. So knowing the genome is not enough. There are issues not captured by the genome directly. Epigenetics is the key word for that. There are questions that have to do with how proteins are expressed. That has to do with regulation. That is beyond the proteins. That's an issue. And then there's the microbiome. Don't forget, most cells that you see walk in front of you are not human. So if you move from your chair, most cells that stand up are bacterial. A number of cells, not size of cells. Most of the material that stands up is water. No, but it's most of the material is human cells, but in weight. But in terms of number, most cells are bacterial. Anyway, so there's a bunch of things that are not captured here. But let's get back to the question: could we have a drug tailored for me? And that brings me to the next question: how many drugs are developed every year? New drugs, something we don't have. Not something replacing with a different label but the same chemistry inside. Generic solutions for something that had a patent and is run out or something like that. That's not what I mean. Or copycat drugs. But really something new. Yeah?
Around 200 something.
Does anybody believe him? 200?
Yes.
Does anybody believe these guys? So here's the answer. Again, this is not up to date, but the number has not changed much. This is US only, but FDA, but essentially is representative for the world. 40 per year. So we were totally right. Fewer than 100. And why is that, yeah?
Is this because it's so long to develop a new drug, or they're required to test it for like 20 years?
Wait, 2 points. I'm going to address both points in a moment. First of all, on average, it takes 14 years. On average, this is outdated now. These numbers are higher. Meanwhile, on average, when I pulled this slide out, and 6 years ago or so, 2 years after that was done, it was still true that it was 2 billion. At this point, the number stands almost at 4 billion. So it takes a long time. Now back to the question that you have. About half of this here goes into the phase of testing. Half of it, so 7 years in developing the drug. So if you imagine a country in which every drug that is developed by the pharmaceutical is put into a person, no matter whether they die or not, right? Or if you are somebody who can afford, if you're the millionaire or billionaire who can actually afford, I have a disease, I can afford the 2 billion, I pay for the drug, but I still have on average to wait 7 years. At least, right? This has not changed. And in fact, this number is going up despite all the advances in biotechnology. And again, the question can be why. Partially the answer is because FDA approvals have become more complicated, but that is not this number that is going up. This is not the half. The reason why this goes up essentially is because we're targeting, initially 20 years ago, pharmaceutical companies took the low-hanging fruits. So the things we're going for are more complex drugs now, and the story is just becoming more complicated. That's as simple as that. We hope that this will all change with CRISPR, or that CRISPR will make a big dent into the image. For the time being, this is the reality on the ground. That, by the way, also explains why pharmaceutical companies are so large. Because if you need one of these big selling drugs, otherwise you're a gunner, and only very, very big companies can afford to have several losers. If for a loser you pay so much money. And that actually makes them bigger and bigger. There's no other system underneath than this mere fact explains the growth, yes?
Is it possible that if you're like a multi-billionaire guy and you want to just discover some drugs to speed up this, like you spread the budget to multiple institutions?
But that's what they do, drug companies. And this is again, so one result, not the reason why pharmaceutical companies merged, but one result of the merger was that all the big drug companies out there are spread all over the world. And typically they are, the way they grew historically, they are spread in different places with local factories of local different expertise. So they actually do different things. So they do that already. And there is a lot of sort of maybe you can optimize it, but actually they tried many phases of optimize. It's really a really tough game. Okay. And there's a variety of reasons why. But anyway, so the reason why I bring this is only to make it clear to you, this genome reading to developing my own drug, no matter what money I have, is unlikely to happen. There may be some examples that you read in the press, but this is not going to be the story. Not only because not most of us are millionaires. I don't know, in New York there are 200 billionaires. So we could have some, but still, the time issue remains the issue. Now, what that means is that ultimately reading the genome will not lead to different drugs. It will lead to different diagnoses. It will lead to different ways of combining drugs. And in fact, that is what we see already. We see that more and more drugs are repurposed, combined. You have cocktails. HIV is one example where only cocktails help. And ultimately, you have things that are already on the market and you combine them in different ways. Diagnosis, choice of food, choice of drugs. That is what will be influenced, or is already being influenced, by reading your genome. For people who carry rare diseases, that happens today at Rechtsisa. And it happens more and more. Today, the way they do it is they look up your genome. You're carrier of a certain SAF that is known to sort of have a problem with a certain drug. You don't get that drug, or you have to take another one. That already happens. And there will be much, much more of that. Again, we will somehow curtail this through our lifestyles or through doing it differently. Our genetic differences matter, and I believe they matter because this is the way evolution works. So what is bad for me, maybe this particular variant is not going to be good for me, but for the group, for human, for the species, this is trying it out, because maybe one of my ancestors will survive better due to that mutation. And who cares about me, as long as I get the ancestor? The moment I get the ancestor, I can die. So my happiness with my problems is not relevant for the survival of the species. So in some sense, I'm selfish enough to call that bad, right? Because I want to be happy too. And in some sense, what is bad for us may be good for the species, but maybe we can sort of curtail the bad effect, and that ultimately would somehow allow me to curtail the bad effect, and that would allow me to sort of benefit from the diversity, because we survive through our diversity. That is why we have this diversity. That's why it's created. Bad effects are not created because of an advantage, because diversity is the way we survive evolutionarily. And in this sense, we would sort of combine diversity and what is good for me. And for me, this is sort of the combination of two different aspects of best of two worlds, let's put it like that. For that, however, we need a molecular understanding of disease, and we are far away from that. What we are doing today is this sort of thing. We want to distinguish between lemons and really good cars. I like that car. And in order to do that today, what we tend to do is we say, okay, I have a bad image here. Get yourself a white car. Because a white car has fewer time in the garage, simply because it's white, you can see it better, and because you can see it better, it survives longer. Doesn't mean it's a better car, right? We're not understanding the way we do that today. We're not understanding really the function of disease. We understand outward signs. And this is sort of, we have to go through a cycle where we really understand the impact of variants on the molecular effects and how we can cure that. And we need several cycles of that. And there are many, many aspects of predicting protein function that are needed in order to improve that, and relating our predictions directly to disease, to the bedside. Many people are working on it. Maybe some of you will begin to work on that. This is just sort of my experience from giving a TEDx talk at some point that clearly changed the way I saw giving talks, and it was really tough for me to learn to do that. It is online, this TEDx talk. So